3, 2, 1. Okay. I don't know how to do it. Oh no. We're just going to go with it. Hi, sorry for that 30 seconds of your wasted time. We are now going to discuss lipid metabolism. So, we have learned how carbohydrates play a very important role in producing and getting to acetyl-CoA so that we can then go to the Krebs cycle and then on to the electron transport chain. But how do fats, blue is the worst color, how do fats undergo aerobic metabolism? And that's what this lecture is going to discuss. So, fats or lipids are available to the body from 3 sources. Number 1, they can be ingested from the diet. Gee, wow, I am off my game. Ingested from your diet. They can be stored in adipose tissue. Of the body. And 3, they can be synthesized in the liver. So hopefully that's not new information as we've discussed some of these concepts already, but this is how fats are available in the body. So for aerobic metabolism, when we're talking about fueling prolonged low intensity activity, we are mainly talking about fat being the primary source. So fat supplies more than 2 times the energy of carbohydrates, and the ability of muscles to use fat for fuel depends on the intensity of exercise. So we're really talking about that low intensity activity. So running at a very doable pace for hours on end, training for that marathon, doing a crazy 50-miler race, but you're really at a low intensity, but you're continuously moving. And so that's when you're going to use your fat as fuel. So again, here's another sort of simplified graph, but let's go ahead and break this down so we really understand what we are looking at. So on the x-axis, we have low intensity exercise going to high intensity exercise. And then on the y-axis right here, we have fuel used. And in this particular case, we're talking about either carbohydrates or fat. So if you are at a low exercise intensity, that means that you are burning 100% of fat. If you are at a high intensity, that means that you are burning 100% carbohydrates. So if we have a long duration or a short duration, that is going to depend on what fuel we use for exercise. And in the coming lecture, we're going to talk about this really cool crossover point. But for now, I really want you to understand that long duration, low intensity exercise is mainly using 100% fat, whereas shorter, high intensity exercise is going to be using carbohydrates as fuel. So back to fats, triglycerides. So this is our chemical name for fats. Triglycerides are the primary form of fat in food, and triglycerides combine with glycerol to supply energy to the body. So we know that triglycerides are fats. Here are some examples of triglycerides, are fats and oils. We know that there are better sources of fat, like the unsaturated fat versus the saturated fat that tend to be not as healthy for us. So foods high in fat are fatty meats and fish, avocado, cheese, nuts and seeds, chocolate, and butter. And so your body is able to utilize all of these types of food for fueling these long duration, low intensity exercises. So let's go ahead and get into the structure of a triglyceride. So we know that triglycerides are broken down to form fatty acids and glycerol. So the backbone here is glycerol, and triglycerol means 3 fatty acids. So this is the structure of a triglyceride. You have a glycerol backbone, and then you have 3 fatty acids. So we also have what is known as digestive enzymes like lipase. And so what lipase does is it essentially breaks that bond, that backbone of the glycerol and fatty acids, to where you just have glycerol and then your 3 fatty acids. So these triglycerides are broken down to form fatty acids and glycerol. So when we are talking about lipid metabolism, we are talking about the breakdown of fats into cells, of lipids, fats in cells, or the breakdown of storage of fats for energy. So these fats are obtained from consuming food and absorbing them, or they are synthesized from fats stored in the liver. We've already established that fact. So we have 2 things going on here. We have ingestion of fats, and we have storage of fats. So this is sort of the same visual. This picture is the same as this, but we just have a little bit of a different visual here. So let's go ahead and look at this. So we've already discussed our carbohydrate metabolism, and now we are discussing our fat metabolism. So as you can see here, we have fats that when they're ingested, we break down the fats, so lipolysis, breaking down of fats, in order to be converted to acetyl-CoA so that we can go into the Krebs cycle and the electron transport chain. Or we have the creation of fat, lipogenesis, from acetyl-CoA that will also help us be able to get to the mitochondria and eventually produce our ATP. So for our lipid metabolism, we know that lipid metabolism begins in the intestine, and this is going to be where ingested triglycerides are broken down. And they are broken down into smaller chain fatty acids. And this is done by pancreatic lipase. So this is our pancreatic lipase that breaks this bond so that you have your glycerol and then your 3 fatty acids. So together, pancreatic lipase and bile salts break down triglycerides into our free fatty acids. And so from this, we have lipolysis, which happens in the cytoplasm. And essentially, to obtain energy from fat, triglycerides must first be broken down into 2 main components: our fatty acids, which will undergo beta oxidation, and glycerol. And this takes place in the cytoplasm. So the resulting fatty acids are oxidized and they're broken down by this beta oxidation into acetyl-CoA. And now this can be used to go into the Krebs cycle and the electron transport chain to eventually produce this ATP. So triglycerides yield more than twice the energy per unit when compared to carbohydrates. Therefore, when glucose levels are low, when you've depleted your they're low. You've depleted a lot of your glucose levels. When they're low, triglycerides can be converted into acetyl-CoA and used to generate ATP in aerobic respiration. So again, we have lipolysis with our beta oxidation, and this is our breakdown of fatty acids. And these fatty acids are converted into acetyl-CoA molecules. I'll spell it right, probably never. Acetyl-CoA molecules. And these fatty acids are then transported across the mitochondrial membrane Remember, we want to get into the mitochondria. So through a series of reactions, our fatty acids are converted to acetyl-CoA, and the newly formed acetyl-CoA can enter the Krebs cycle. And this will be used to produce ATP in the same way that carbs are from pyruvate. So again, I'm going to go back to this right here. So we just talked about this fatty acid spiral. This is our lipolysis to get to acetyl-CoA, which then will allow us to get to the Krebs cycle and the electron transport chain. So that was lipolysis. Now, we do have another way that we use fat as energy. And so that will be lipogenesis. So when you eat more glucose and carbs than your body needs, your system uses acetyl-CoA to turn the excess into fat. So acetyl-CoA initiates lipogenesis. And this formation of fat usually will occur in the cytoplasm of liver cells. So if you break down the word lipo, which is fat, and genesis, which is the creation, we are literally saying here the creation of fat. So this conversion of carbs to fatty acids, which is what this picture here is depicting. So for our lipolysis versus our lipogenesis, remember that lipolysis is the breakdown of triglycerides into fatty acid and glycerol, so the breakdown of fat, whereas lipogenesis is going to be this creation or this formation of fat. So we have this concept in physiology and exercise physiology in particular that discusses this interaction of fat and carbohydrate metabolism during exercise. So glycogen is depleted during prolonged high intensity, I don't know why it keeps erasing on me, high intensity exercise. And so this causes a reduced rate of glycolysis. This also means if we have a reduced rate of glycolysis, we have reduced intermediates for the Krebs cycle, which then means we will have reduced fat oxidation. And we know that fats are important for the Krebs cycle and that they're metabolized by the Krebs cycle. So we really do have this nice interaction of fat and carbohydrate metabolism during exercise. So the depletion of carbs reduces the rate of glycolysis. Therefore, the concentration of pyruvic acid in the muscle is also reduced. So this is going to reduce Krebs cycle intermediates, which then in turn will decrease the production of our ATP. So when the rate of glycolysis is reduced, that means that we limit ATP production. So when carbohydrate stores are decreased and depleted in the body, the rate at which fat is metabolized is also reduced. So it's important to understand the interaction of fat and carbohydrate metabolism during exercise. So our final graph here today is looking at the effect of exercise on duration and muscle fuel source. So again, a lovely graph. Let's orient ourselves here. So on our x-axis, we have exercise duration in hours, 1 hour, 2 hour, 3 hours, 4 hours. And then on our y-axis, we have percent energy expenditure. Usually when you see percents, that's going to go up to 100, and this graph does as well. And so over on the right-hand side, we know that carbohydrate is stored as glycogen in liver and muscles. And this muscle glycogen stores provide a direct source of carbs for muscle energy and metabolism. And so I'll let you sort of read some of these key concepts on the right-hand side of this. But what I want you to look at is that for this 1 hour of exercise time, we are relying on about 45% of our muscle glycogen. Maybe about 5 to 10% of blood glucose, about 25% of plasma-free fatty acids, and then about another 25% of muscle triglycerides. So as the exercise duration and time increases, look, our muscle glycogen stores become less, whereas our plasma-free fatty acids, we tap into those stores a little bit more. So being able to read this graph will serve you well. But this lecture was about fat metabolism and how that plays into our aerobic respiration. See you on the next one.